Chapter 12, Section 2, DNA Replication. When Watson and Crick discovered the double helix structure of DNA, there was one more remarkable aspect that they recognized immediately. The structure explained how DNA could be copied or replicated. Each strand of the DNA double helix has all the information needed to construct the other half by a mechanism of base pairing. Because each strand can be used to make the other strand, the strands are said to be complementary. If you could separate the two strands, the rules of base pairing would allow you to reconstruct the base sequence of the other strand. In most prokaryotes, DNA replication begins at a single point in the chromosome and it proceeds, often in two directions, until the entire chromosome is replicated. In the larger eukaryotic chromosome, DNA replication occurs at hundreds of places. Replication proceeds in both directions until each chromosome is completely copied. The sites where separation and replication occur are called replication forks. Duplicating DNA. Before a cell divides, it duplicates its DNA in a copying process called replication. This process ensures that each resulting cell will have a complete set of DNA molecules. During DNA replication, the DNA molecule separates into two strands and then produces two new complementary strands following the rules of base pairing. Each strand of the double helix of DNA serves as a template or model for the new strand. Figure 12-11 shows the process of DNA replication. The two strands of the double helix have separated, allowing two replication forks to form. As each new strand forms, new bases are added, following the rules of base pairing. In other words, if the base on the old strand is adenine, thymine is added to the newly forming strand. Likewise, guanine is always paired up to cytosine. For example, a strand that has the bases TACGTT produces a strand with the complementary bases ATGCAA. The result is two DNA molecules identical to each other and the original molecule. Note that each DNA molecule resulting from replication has one original strand and one new strand. How replication occurs. DNA replication is carried out by a series of enzymes. These enzymes unzip the molecule of DNA. The unzipping occurs when the hydrogen bonds between the base pairs are broken and the two strands of the molecule unwind. Each strand serves as a template for the attachment of complementary bases. DNA replication involves a host of enzymes and regulatory molecules. You may recall that enzymes are highly specific. For this reason, they are often named for the reactions they catalyze. The principal enzyme involved in DNA replication is called DNA polymerase because it joins individual nucleotides to produce a DNA molecule, which is, of course, a polymer. DNA polymerase also proofreads each DNA strand, helping to maximize the odds that each molecule is a perfect copy of the original DNA.